Okay, good morning. Welcome to VSPG today. We're going to hear from Dr. Akshay Mehra on Precambrian life reconstructed. Next week, we have a presentation by Felix Gervais from Montreal Polytechnic. And I'll send the email about that on Monday. I will go ahead and introduce Akshay for us and we can get rolling. So Dr. Akshay Mehra is a paleontologist and sedimentologist who uses quantitative descriptions of form and space to better understand the evolution of life and the environment on Earth. Paleontologically, his work is well established in studies focusing on Cloudina and Namapoikia, but he also applies his work towards better understanding of sedimentary structures, and recently even on crystal settling in magma chambers. Akshay received his bachelor's degree from Cornell University and his PhD from Princeton, working with Dr. Adam Malouf. His thesis there was titled Reconstructions of Ediacaran Putative Biomineralizers via a novel serial grinding and imaging technique. After his PhD, Akshay completed his postdoctoral fellowship at Dartmouth College, working on Paleozoic reef structures and geochemical sedimentary record reconstructions before starting his current position as assistant professor at University of Washington. Akshay, this is going to be a really cool talk, so I'll leave it to you.
Awesome. Thanks so much, Alex. Thank you all for coming and for inviting me to give this presentation. As Alex mentioned, I actually work on a whole range of questions. And in fact, now a lot of my research involves rocks that are significantly older than the Ediacaran or significantly younger. But I'm really psyched to be telling you the story about Cloudina and Namapoikia and these Ediacaran putative biomineralizers. So before I continue, I just want to point out that this photograph is of a bedding plane taken on Saline Mountain in the Canadian Rockies. It's a set of trace fossils, and this bedding plane sits stratigraphically below bioherms that contain Cloudina and Namapoikia. We are at some point in this talk going to return to Saline Mountain and then return to those bioherms and ask what those Cloudina are actually doing there. But before I get there, I really want to start with a much broader statement, which is the misidentification of Precambrian life has been a challenge for more than 150 years. There was some dialogue in the chat before we even started this presentation talking about pseudofossils. And it turns out that for the last 150 years, we've really struggled to understand whether a fossil is biotic or not. And even once we understand that, we struggle to understand what those organisms were doing in the places in which we find them. This problem goes all the way back to Darwin. So when Darwin published Origin of Species, he had to contend with the fact that there were no known examples of Precambrian fossils. Darwin put forward this idea that if his theory was correct, then the world had swarmed with living creatures well before the Cambrian radiation, and that eventually we would find examples of these Precambrian of these Precambrian organisms. And almost immediately after Darwin published Origin of Species, one candidate fossil emerged. This is Eozoon canadense, the dawn animal of Canada. It was discovered in 1858, but really publicized and accepted as a Precambrian fossil in 1864. And Eozoon essentially comprised a laminated construction of alternating calcite and serpentine bands in most cases. Eozoon appeared to be the response to Darwin's so-called dilemma, and really careful microstructural and microscopic work illustrated that there were these canal systems within these fossils. Others came out with these sort of putative reconstructions demonstrating what Eozoon was doing, how it was growing. And the idea was that Eozoon was essentially a primitive form of foraminifera. Now, it's important to note that even as Eozoon was introduced as a biotic remnant, people were pushing back against that idea, and an acrimonious debate actually ensued for decades. And that debate was finally put to rest through two different studies. The first was a set of comparative analyses comparing a sample of Eozoon, you can see that on the right-hand side of this drawing, to potential analogs of modern foraminifera that Eozoon was said to represent. And so these comparative analyses demonstrated that there was little resemblance between these two kinds of fossils. And then there was a second study that essentially found similar looking forms in volcanic ejecta. So on the foothills of Mount Vesuvius, here's a plate from this specific paper by Johnston, Levis, and Gregory. And here, the whole idea of Eozoon being a biotic organism was put to rest. So the reason I tell this story is because I want to point out that if we're going to decode the history of life on our planet, and especially as we deal with enigmatic Precambrian forms, what we're going to need are multi-pronged studies. So we don't just need quantitative analyses or qualitative potential reconstructions, but we also need comparative observations. And all of these things working together will help us determine whether something is biotic or not. And then more importantly, if it is biotic, what impact it had on, say, evolution or on the paleoenvironments in which it lived. And these sort of analyses are incredibly important because today, Precambrian fossils or examples of Precambrian fossils abound. This is an excerpt of a figure by Hans Hofmann from 1971. This is in a book about Precambrian fossils from Canada. It's only the top half of the figure, and all Hofmann is doing here is laying out the descriptions of fossils as they've appeared in our recent time. And already what you might notice is that some of these fossils have been marked as problematica or have been shown not to be biotic at all. I think it's probably useful now to just explore what we know about life up to this time period, up to the Precambrian-Cambrian boundary. And then I'm going to drill in on those Ediacaran biomineralizers that I want to tell you a story about. So here I'm just showing a timeline going from 0 MA to 4.5 GA. In yellow, I've marked the boundary between the Precambrian and Cambrian, and I've marked the eons here. And if we want to go all the way back to when we have the first evidence of life and walk right up to this Precambrian boundary, we'll begin with microbes, or we would begin with microbes and microbially mediated construction. So stromatolites and thrombolites, which are either laminated or modeled microbially mediated constructions. Here's an example of a modern stromatolite from Shark Bay in Australia. But essentially, these constructions are probably the only evidence of life we have for billions of years on our planet. There are examples of trace fossils about 2.1 GA, this specific example, but there are other examples put forward. And the idea behind these trace fossils is that we know that there is at least some motility. Now, what the nature of that motility is and what the organisms were that actually made these trace fossils remains an open question. As we move further up towards that Cambrian radiation point, towards that boundary, we start to see the last common ancestors of eukaryotes. And there are many eukaryotic microfossils. So here's an example. This is from a paper by Cohen and Kodner. And essentially, people who are much more skilled than I are able to extract these microfossils from a whole host of Proterozoic rocks. I want to point out that these fossils themselves are also enigmatic and interesting and challenging to decode. As we get closer and closer to that boundary, we start to see evidence of what might be metazoans. This is a paper by Turner from 2021 that suggests that these keratose fabrics, so you can see in panel A, are actually the remnants of sponges that are coexisting in a microbial system. And Turner's hypotheses and interpretations are actually based on work that's done by Lee and Writing that demonstrates that, at least in the Cambrian, we start to see consortia of both stromatolites, so these microbially mediated constructions, and then sponge fabric. So here on the left are a set of stromatolites from upstate New York. On the right side are zoom-ins that show those same keratose fabrics that Elizabeth Turner presents. Now, as we get even closer to that boundary, we start to see the Ediacaran biota. So people in this room might be very familiar with Ediacaran biota. But the idea here is that these are soft-bodied organisms that leave behind incredibly fine impressions, and that the nature of these biota are still heavily debated. So if you go back to the 90s, there's this idea that essentially these were some form of failed experiment on the part of nature, some sort of pneumatic construction that existed and then went away. We know from certain biomarker data today that at least some of these Ediacaran are metazoan in nature, but there's a lot of work that is being done and needs to be done on these Ediacaran to really understand what they're doing. and what their biological affinities were, etc. I'm most interested in this moment right before the Cambrian, when we start to see putative biomineralizers. So there are many different kinds of putative biomineralizers. There are at least 4 genera. I'm showing you 3 here and the 3 that we're going to focus on for this talk today. There is on the left-hand side Damocalathus. This is a polished slab, and all I've done is drawn a yellow line around a single individual. There in the center is Cloudina. This is a photograph of a bedding plane from Namibia. And Cloudina is found in these very large aggregates over the world. And then finally, we have Namapoikia, which is this labyrinthian construction found in a single reef in Namibia and is thought to be a putative sponge. And there are 2 major questions I was interested in, am still interested in, and I think many people should be interested in. The first one is, what are the biological affinities of these early biomineralizers? And then secondly, what were these things doing in the settings in which they're found? So in all those shallow water settings, what were the paleoenvironmental impacts of these organisms? And then perhaps more importantly, from a broader scale, what were the evolutionary impacts of these organisms? And today I'm going to make the case that 3-dimensional reconstructions are one of these really detailed analyses that we need to do in order to answer those questions. And in order to convince you of that point, I'm actually going to walk through a description of why 2-dimensional projections don't really do everything we want them to do. And I'm going to begin with William Sollas, who was a geologist at the turn of the century. And in 1904, Sollas made a presentation to the Proceeding of the Royal Society, where he essentially said that geologists and paleontologists were at a disadvantage when compared to biologists, because biologists could go ahead and produce serial sections. And what Sollas was talking about was, if you're a biologist with a very sharp knife and you have some sort of soft tissue, you can produce incredibly thin sections through that tissue. And you can lay those sections out in order, and then you can understand how different features within that tissue relate to one another. So for example, here we're looking at serial sections through a rat brain. If you've ever been to a bodies exhibit, you know what serial sections are. And Sollas essentially lamented that in the case of many fossils, not a half, frequently not a tenth of the information they could afford had been extracted from those fossils because they were embedded in rock and all we had were 2-dimensional projections to work with. And I think one reaction to that statement is to say that's hyperbole, because we know we can extract really meaningful information from thin sections and 2-dimensional projections. But I'm going to make the case that Sollas was really onto something, and it's something that we need to be very concerned about. I'm going to make that case by looking at archaeocyathids. So now here, we're no longer in the Precambrian, we're actually in the early Cambrian, the mid-Cambrian. And archaeocyathids are calcareous sponges that are thought to be Earth's first framework reef builder. These things built topographically diverse constructions and really were producing these dense systems, much like corals today. So Ryan Mansick, who's currently a graduate student at Princeton, has been working on quantifying archaeocyatid growth. And he and I were out in Nevada one year looking for these archaeocyathids, and we came across this bedding plane. I apologize for the slightly blurry nature of the photograph. But if you're someone who's looking for these calcareous sponges, you might find examples of them in this bedding plane. I've highlighted 3 here with dash white lines. If you have been looking at archaeocyathids for a long time, as Ryan has, or as I had at the time, you might see this branching figure here. So you now know from this single bedding plane that there are multiple of these archaeocyathids within a small region. We know that at least some of these archaeocyatid sponges seem to be branching. But what we miss when we look at all of this are the density of construction, the sort of density of how many archaeocyathids are there. We're unable to reconstruct the branching angles, the 3-dimensional branching angles of these organisms, and we're really unable to compare them to modern corals from those single projections that we have. And at the very end of the talk, I'll show you some more work that Ryan's been doing with these archaeos to demonstrate that, in fact, they do have traits very similar to modern corals, and they were able to change the flow around them, or at least adapt to that changing flow. Something that many people might be familiar with, and something that I learned through doing synthetic cross sections. So essentially we build 3-dimensional models, computational models, and then we just cut them randomly. And then we look to see what our estimate of certain properties are in those 2 dimensions and how they compare to the known 3-dimensional values is that three-dimensional properties cannot accurately be estimated from single 2D cross sections. Here I'm just showing you a set of those synthetics. So we might go from looking at tubes and what happens when those tubes are curved or bent, or if they're not perfectly circular in cross section, all the way in the bottom right to looking at single minerals. So if we're trying to say something about pseudomorphs, we have to understand if we're looking at a single 2D bedding plane, we might be completely underestimating what kind of symmetry we have. And the point I want to make here isn't that all two-dimensional projections need to be thrown out the window. The point I want to make here is that we need to understand the error that emerges from these two-dimensional measurements. We have to understand the magnitude of that error, perhaps by going ahead and ground truthing, or perhaps by building synthetics and doing random cross sections. And that we have to understand whether those errors are tractable. And if they are, we can proceed ahead with whatever we're doing. So one example of this is on the lower left of this figure, which I'm showing you a granular material. And if we take that granular material and we make a cross section randomly through it, and we look for porosity, so we're just trying to estimate what the porosity of that volume is, we're going to come within 2 to 5% of the true porosity. And if that 2 to 5% error is much lower than the signal that we're searching for, then those two-dimensional measurements are perfectly valid. So I'm not saying throw out two-dimensional measurements. I'm saying that sometimes those errors might be intractable. In that case, we need three-dimensional reconstructions. Now, I'm going to return to Solis, and I'm going to tell you that once Solis was done lamenting, what he did was build this machine. This is a serial grinder and imager. Well, it's just a grinder, actually. So what you do is you take a sample and you mount it over in E. And then what you do is you turn this wheel and slowly you grind away a little bit of material. Solis did about a millimeter at a time. You take that freshly ground surface and you make a tracing of it, or perhaps a photograph. You have now a projection you can work with. And what you can do is you can start to stack those projections. This is what Solis and his daughter did with a set of fossils, and you can stack those projections and reconstruct the three-dimensionality of the fossil that was once embedded in rock. Something I find especially cool is that what Solis did was actually take these projections and then assign a thickness associated with the amount of material that was removed, and was able to build wax models of these fossils. So essentially deleting the rock around it. And these are really just analog forms of what I do today. So I think these are just absolutely fascinating historical artifacts. But two things that might emerge from this story is that it appears that this is really time consuming, and more importantly, that this method tends to destroy the samples that you're after. Luckily, today, we don't have to necessarily destroy samples. There are non-destructive tomographic methods. Many in the room might be familiar with X-ray computed tomography. And the idea here is that you take X-ray beams and you shine them through a sample. Those X-rays are attenuated on the basis of the materiality of those samples. And what ends up happening is if you rotate that sample, you start to produce multiple projections. If you use inverse methods, you can reconstruct the three-dimensionality of the features within that sample. And the question might emerge, okay, so if we have tools like X-ray computed tomography or other non-destructive methods, why might we still use Solis's method? And I'm going to tell you all tomographic methods, including serial sectioning, rely on some sort of contrast. In the case of serial sectioning, we're relying on optical imaging and differences in the images themselves. So for example, when we're looking at a granite, the two feldspars look different in color and texture than the mafic elements and then the quartz. And the same when we're looking at a carbonate. For example, here we're looking at a Cloudina bearing sample. So the Cloudina in white, we have some cements in white as well, but then the matrix is a dark gray micrite. And we also see a little bit of dolomite there in yellow. So that contrast really pops when we look in optical imaging. But for example, if we go look at X-ray CT, we see a different story. The oolite here, so oolites are just rocks made up of ooids, which are concentric carbonate grains. Those oolites, the ooids show up very clearly here, and that's because there's a clear density contrast between those carbonate grains and then the air around them. When we move to granite, what we end up seeing is that the feldspars and the quartz actually have similar densities, and so those really disappear in this X-ray CT. And by the time we get to the carbonates, we almost get no returns whatsoever. there are people out there and there are labs that do exceptional work with X-ray CT and extracting boundaries in even these low density samples. So Jim Schiffbauer's lab at Missouri and people in his group come to mind. But the point I'm making with this slide is that there's still room to be using serial grinding and imaging. There is still a need for destructive analysis in certain cases. And what that need for destructive analysis has led to is the construction of the grinding, imaging, and reconstruction instrument. This is something I worked on when I was a graduate student. I'm attempting to build one here at the University of Washington now. Essentially, it comprises a CNC grinder, so a computer numerical control grinder, just this really big surface grinding machine. And then what we've done is attach a high resolution camera. It was 80 megapixels, now it's 100 megapixels. In addition to that, we have a multispectral imaging setup now, so we can image all in UV all the way to near infrared. And the way this system works is essentially we take a sample, we mount it onto a plate, we then grind away a certain amount of material. This is all automatic. That ground surface moves underneath the high resolution camera. We take a photograph and we essentially mimic what Solis was doing, removing a certain amount of material at a time, often 30 microns at a time, until we've destroyed the sample, but we're left with a stack of high resolution images that are on the order of about 5 microns per pixel in resolution. And so this method here is exciting to me because it opens up new opportunities for reconstruction. Not only can we image samples that are somewhat opaque to non-destructive methods, but we can also image very large volumes. So here I'm just showing you spatial resolution versus the amount of volume that we can analyze for a series of sectioning methods. So we go from 3D atom probe in the bottom left, all the way up to Geary in the top right. And so a lot of our three-dimensional reconstructions through serial sectioning actually occur in the nanometer to micron range. But here with Geary, we're actually working in the micron range of resolution, but the millimeter to centimeter scale. So we can image volumes that are 10 centimeters by 10 centimeters by 10 centimeters. So relatively large volumes at relatively high resolutions. And to make the case that Geary is not just meant for fossil reconstruction, I'm going to just make a quick aside and show you these reconstructions of ooids that Bolton Howes has been working on, or it's been spearheaded by Bolton Howes, who currently is a postdoc at the University of Miami. And so one thing we were interested in is how the shape and size and the shape evolution of ooids, so how they go from being elliptical to spherical or vice versa, can tell us something about paleohydraulic and paleoenvironmental conditions. And as all you might be familiar with, there are many open questions about what the size and shape of ooids tell us about those conditions through time. This specific example is a flip book, so we're just going through photographs of giant ooids. So giant ooids are just anomalously large ooids. And we're just moving through the stack and we're just seeing how these shapes evolve. Of course, like anything else I've shown you, two-dimensional projections have a lot of errors. And so if we're interested in shape evolution or size evolution over time, we need to be concerned if what we're working with are two-dimensional projections, so thin sections. Here are just 3 examples from a paper that Bolton led in which we examined the errors that are associated with 2D projections. On the left-hand side, we're looking at what happens if you take a ideal ellipsoidal form and you take synthetics of those. That's in the inset there. You take a cross-section and you measure the major axis length, so the longest length in that cross-section, and then you compare that to what the known major axis length is of that ellipsoid. And clearly what you can see is that if you're doing that, you're going to underestimate the true major axis length. In the middle panel, what we're looking for, we're showing the errors associated with major axis length up here on the left-hand side, with the 2 other axes, so the B and C axis. On the lower right, we're showing what happens if you're looking for aspect ratios. So if you're trying to understand how circular that cross-section is, you can get errors of up to 50%. Finally, on the very right, what I want to demonstrate here is that one answer to this issue with projections is that, well, we can just take more projections. What ends up happening when you take more projections isn't that you actually reconstruct the true value, but you converge on an answer that is wrong. And that convergence, your estimate shrinks in the confidence interval, but isn't actually anywhere close to the true value, which is in that red line here. And one thing Bolton showed in this paper, essentially, is that actually when we start to think about growth trajectories, we have to be very concerned if we're just looking at 2D cross-sections. So here what Bolton has done is taken a true three-dimensional reconstruction made with Geary, that's shown on the bottom right. So taken a single ooid and reconstructed the different levels of its growth. And then what he's done is taken 20,000 cross sections through this one ooid that we know of, making sure each time to at least cut through the cortex. And then he's looked to see how the sphericity changes across that cross section. And so the delta sphericity here just says, what is a change in sphericity from the very center all the way to the very edge of this cross section? So in the top, what we see is that ooid actually appears to become less spherical as it grows and then it returns to some sort of spherical nature. The other cross section, the one shown in the middle, is actually never achieving true sphericity. And neither of those values are actually anywhere close to what we know the true delta sphericity is. The point to make here is that if we're trying to say something about growth history through time, we need to be very concerned about those 2D projections. And a lot of the 3D work that Bolton did actually has led him to start to consider how we might describe ooids in different ways, the three-dimensional forms. And so here is just showing how we might look at spherical harmonics to look at the differences in ooid generation from ones that experience no abrasion to ones that might be experiencing collisional abrasion to ones that might be rolling, and how the spherical harmonics differ there. So if you want more details about this, you're going to have to ask Bolton or ask him to present, but just be on lookout for this work. It's in review now. Okay, that was a long aside to get back to the Ediacaran putative biomineralizers, and what I hope I've convinced you of is that we need to worry about three-dimensional reconstructions if we're interested in understanding what these organisms were doing in the places in which they're found. So now we're just going to step through these three different organisms. We're going to start with Namaqualathus. I'm going to tell you that Namaqualathus has been reconstructed in 3D by John Grotzinger and Wes Waters in 2000s. This is what their reconstruction looked like. Essentially, it's a goblet-shaped organism that is relatively flexible. More recent work by Shore and others has demonstrated that there is at least some pyritized soft tissue in there. So these are three-dimensional reconstructions using X-ray CT of an exceptionally preserved sample. And the idea here is now we know Namaqualathus to be a Lophoturcozoan. The actual growth habit is still something that we might consider and think about. And there's some interesting work to be done there. But I haven't reconstructed Namaqualathus. I have some ideas about that. If anyone's interested, I'm happy to talk in more detail. But what I'm actually going to do is focus on Cloudina and Namapoikia, as Alex mentioned at the very beginning. And we're going to begin with Cloudina. Once again, this is a bedding plane photo, and this is actually pretty representative of how you find Cloudina in bedding planes across the world. So you go to a shallow water reef and you find them in very, very large aggregates. So a reasonable question that might emerge is what role these Cloudina had in building the reefs in which they're found. And work by Penny and others in which they looked at bedding planes of Cloudina and measured the orientation and observed cementation between individuals. And this orientation suggested at least these Cloudina appeared to be oriented in a single direction, consistent with orientation in a current to do with feeding. So the authors put forward this idea that actually Cloudina and not archaeocyathids were the first framework reef builders on our planet. And so why does it matter whether Cloudina are building reefs or not? Why should we care? Why should we test this hypothesis? For that, I'm just going to introduce this really basic concept that for most of Earth's history, reefs are the domain of microbes. So microbes are building amazing constructions in these reefs, but those microbes are producing constructions that are morphologically distinct from reefs that are built by calcifying metazoans, what we would call framework reefs. So these calcifying metazoans are producing reefs that have high topographic relief. They have nooks and crannies. So if you think about a modern coral reef today, those nooks and crannies act as refugia. Modern reefs are hotspots of biodiversity, and so it stands to reason that they may be the cradles of evolution. Framework reefs might be the cradles of evolution. And so there's a reasonable question to ask here, which is, when did these framework reefs first emerge? And what role did they have in the evolutionary timeline? So was metazoan reef building part of the fuse that set off the Cambrian radiation, or was it a result of rapid diversification? I'm working on these questions actively now, but in this specific case, we're going to look at Cloudina as a metazoan framework reef builder and test that hypothesis to understand whether framework reef building emerged in the latest Ediacaran rather than in the Cambrian with the archaeocyathids. To do these reconstructions, we have to go and actually find these rocks. This is a biohermal or a bioherm up in Saline Mountain. So I told you that we were going to return to Saline Mountain at the beginning of the talk. These bioherms are made up of stromatolites. And if you go and look, if you walk right up to them, you'll find these stromatolites. And within the void spaces or within the areas in which detritus coalesce, you start to find these fossils. They're in white here. So here, all of these little cross sections are cross sections of Cloudina and Namapoikia. Now, the Cloudina, when reconstruct as a set of cone and cone or tube and tube constructions. The ones in Saline Mountain are especially interesting because they have ornamentation. Not all Cloudina have ornamentation. And one thing we can do is we can reconstruct volumes of these Cloudinas, so these aggregates, and try to understand whether there is true orientation or cementation there. And here's a reconstruction from Namibia, from a place where we know, that these Cloudina are coming in within a fill. Something that might pop out to you in this three-dimensional reconstruction is actually there doesn't appear to be a dominant orientation. There doesn't appear to be any sort of size sorting here either. And in fact, in every single example of Cloudina aggregate that we look at world over in 3D, what we find are similar constructions. We find that, in fact, rather than being oriented and organized and in situ constructions, these are all actually detrital inputs. One way we know these are detritus is we can actually look at the cross sections of the tubes. So we can take individual tubes and we can look at their cross sections. If these were truly growing in place, we should expect them to have no deformation. And so they should have circular cross sections plotted on this 1-to-1 line. Every tube we look at is slightly squashed. So it has an elliptical cross section. The true elliptical cross section here is on that dashed line at 0.5-to-1 ratio. And the thing we can also do, because we have three-dimensional reconstructions, is look at the orientation of those cross sections and to see if there's a dominant signal from burial there. And it turns out actually those cross sections are oriented totally randomly. So really the idea here is that this organism these shells are being deformed en route or soon after deposition and before burial compaction and anything else. And so that leads for us to ask this question. We know that these things aren't building reefs, but what are they? Our proposal, or our preferred proposal, is something like a serpulid worm tube. These are agglutinated worm tubes from France, so in a little lagoonal setting. And essentially, if you walk up to these individual tubes, you can squeeze them before your hands and they'll deform easily. They'll in fact collapse. What's really promising from this work is all we had were these deformed body or deformed skeletal fossils to work with. We had no tissue, but Jim Schiffbauer and others were able to find examples of pyritized tissue and reconstruct a through gut, and were able to at least assign a bilaterian affinity to these a few years after our publication came out. So I think this is really exciting. It shows that, yes, we might not have much material to work with when we're working with Precambrian life forms, but that we're able to make, especially through comparative analyses and through quantitative analyses, we're able to make hypotheses that can be tested when we do happen to find certain materials. Okay, so that's Cloudina. We know it's not a framework reef builder. We believe it to be a bilaterian. We believe it to be some sort of annelid, at least personally. Now we're going to turn our attention to Namapoikia, which is a putative sponge. It shows up in the rock record in this single reef system in Namibia. It looks like this labyrinthian construction. It looks ordered. And in order for me to explain why we care about whether Namapoikia is a sponge or not, I'm going to need to tell you a little bit about the Precambrian sponge record. So here we just have this timeline now turned on its side. I've marked where the microbes come in and where we know metazoans should emerge through molecular clock data, through potential biomarker data. And the record of sponges, of Precambrian sponges, is incredibly diverse and incredibly challenged. These are just multiple examples of putative sponges that people have described. And each one of these examples is challenged by at least one other person, if not others. Namapoikia is one of these putative sponges in there. And if sponges are the basal metazoan, we're really interested in when they first emerge. If we're interested in reef construction, we might be interested in when calcareous sponges emerge, because if we think that archaeocyathids are building reefs in the Cambrian, we might want to set off for looking for calcareous sponges in the Ediacaran to see if any of that behavior exists as well. So we're going to test whether Namapoikia is a sponge. And one of the reasons why Namapoikia might be a sponge is because uh, it has morphological features that are very similar to known calcareous sponges. So this is A perforata. This is a sponge from the Ordovician that we know to be calcareous. This is a transverse cross section through the two of them, just a single 2D projection. They look somewhat similar. Here, we are looking at a longitudinal cross section. These two actually look very similar indeed. One thing to note is that the A perforata is much smaller in size than the Namapoikia is. Both of those scale bars are 1 millimeter. So one proposed idea for the way that these sponges grew is that they grew on some sort of microbial substrate. They're growing outwards. And in certain cases, there is cementation on the right side as the sponge grows upwards or outwards. We have partitions, and then we have the presence of tabulae. And the sponge itself is right at the top. As the sponge evacuates the lower areas, as I mentioned, on the right-hand side here, those lower areas become filled with cements, and those partitions tend to become over-thickened. So this is a model for how Namapoikia may have grown. It's based on an extant analog. So this is Vasiletia crypta. This is a very small sponge. That little scale bar there is 500 microns. So this is a tiny little calcareous sponge that exists today. What you might notice are the tabulae that I just showed you and the partitions now in this extant sponge. You might even notice at the bottom that we have over-thickening and calcification. Here's a cross-section through that sponge. And then once again, the sponge is just living at the very top. The living tissue is right at the very top, and those lower areas become filled in. So this is a proposed model for how Namapoikia grew. And for us to actually evaluate Namapoikia as a biological affinity, we need to know something about sponge morphology. So as many of you might know, sponges essentially comprise these cells known as choanocytes. These choanocytes beat in unison. As they beat, they move water over their collars, and they're able to exchange oxygen and nutrients from that moving water. So the most basic sponge just aligns a whole bunch of these choanocytes together, and they beat in unison. As sponges get bigger, they don't actually increase the size of the canal. And you can think about that from a hydrodynamic regime. The bigger that you make this central void, the more stagnant flow you're going to have. So instead, what sponges do is they actually just increase the complexity of the canal system. So as sponges get larger, they're not just growing larger structures, they're growing more and more little canals. And based on all of this information, based on the proposed reconstruction, I'm going to ask this really simple question: is what can we learn about how Namapoikia grew on the basis of a 3D reconstruction? Going into this thinking that this is actually a sponge. Now, when we go look for Namapoikia, we're going to go to the single reef outcropping, as I've mentioned before. It's in Namibia. It's on a farm known as Drudenvlakte. I've marked it in red here. When we go out to the farm and we go look for Namapoikia, we see this sort of fabric. You see this fabric in many places across the reef, but it actually turns out that they're not as ubiquitous as you might first think. One thing that we did when we were out on this reef is do a very careful gridded analysis of the reef itself. And what we're able to produce are maps such as this one. Here, I'm just showing you the probability of finding Namapoikia on this reef system. All you have to know here is that these rocks are dipping 45 degrees to the southeast. And so you have this oblique cut through the reef system. And you only find Namapoikia in areas where you see a hotspot, so where it's red. So there are actually very few points, and they actually just occur in Neptunian dikes within the reef system. And when we go and we collect these samples, we are able to produce the 3D reconstructions. I'm just showing you a fracture so you know what that looks like in this reconstruction. Stratigraphic up is pointed towards us right now. So that's the up direction in which this organism was growing. What we can see from this reconstruction is that these things actually look like a set of sheets rather than canals. There are a whole bunch of giant voids that cut through the system, so they're irregular voids. There's no evidence of tabulae here whatsoever. And so as you cut through this, what we're really looking at is something that looks sheet-like rather than canal-like or chamber-like. Unless you think that this single specimen is giving us a biased view of the sample, we also took a second sample collected by other workers back in the 2000s and reconstructed by Wes Waters at the time, or at least imaged by Wes Waters, and we reconstructed it using our new machine learning methods. And so we have two samples to compare here, and we can just start to ask really basic questions with these samples. The first thing we can do is we can look at the relationship between the thickness of the partitions and the void spaces. So remember what I told you about how as sponges get bigger, they don't necessarily just make bigger constructions. They increase the number of chambers and the density of them. And so what we see here is that when we compare the size of partitions and voids, the histograms of them in sample A and sample B, to a set of known calcareous sponges in the rock record and extant sponges, including V. crypta at the top, all of those sponges sit within that blue bar there. So Namapoikia is anomalously large for a sponge in the rock record. In fact, it's much larger than G. discoforma, which that G stands for gigantic. And so it is the biggest sponge in the rock record, if that is in fact what it is. Then what we can do is we can start to look at the difference in partition and void sizes between sample A and sample B. So these are two samples taken from two different parts of the reef. And we should expect that the partitions tend to be pretty regular and that the voids tend to be pretty regular in distribution. And in fact, what we find is that the voids are completely different. They're off by a whole factor of 2 between these two samples. And that's really strange. So it doesn't make sense why the circulation system of a metazoan would vary by a factor of 2 in two different parts of the same reef. Finally, we can start to look to see if there is over-thickening at the base, as we expect from these models. And in fact, what we find is that in one case, there's actually thinning, so the thickness actually reduces as you go up. That represents over-thickening at the base, although this is a really weak signal. And in another sample, in sample B, you actually have a slight thickening as you go upwards. So it's not very clear whether that property actually exists within these specimens. And to convince you that Namapoikia looks nothing like a sponge, for a calcareous sponge from the rock record, here's a 3D reconstruction we've done of A. perforata. So here what you can see are these really consistent sizes of the canals and of the skeletal elements. In fact, they're incredibly consistent. That makes sense. Metazoans produced really ordered forms. There are no large voids within this reconstruction. So this looks nothing like Namapoikia at all. In fact, Namapoikia is something that we wouldn't even consider to be a sponge, at least in our interpretation. And we're going to argue that it is something more akin to a microbial construction, so something that we're actually seeing aggregation of. And to make this case, I'm going to just show you our preferred model. We essentially have a set of ridges growing in some matrix. So these are the partitions. At time 0, they might look like this. At time 1, we get a split of that ridge for whatever reason. We might have changing nutrient or light conditions. At time 2, we might have a merge somewhere else. And over time, what we're producing is something that looks like a single construction. Our preferred interpretation of Namapoikia would explain why we only find it in a single reef system in the world and why it only occurs within these syndepositional fractures between these Neptunian dikes at reef margins. And that's because there are probably slightly different paleohydraulic conditions and paleoenvironmental conditions at those margins. And so here's our preferred reconstruction for what Namapoikia is. Lest you think that we're out there in totally out there, I'm going to tell you that actually Namapoikia has a potential analog, and that's F. cooperi. This is work done by Russell Shapiro and others. This is a Cambrian thrombolite that exhibits the same sort of sheet-like constructions that both coalesce and merge. Here's a reconstruction of F. cooperi that Russell provided us. We don't know which direction up is here, but we know, so whether up is going top down upwards or from the top downwards. But the point here is what you can see in this reconstruction are the same coalescing and merging that we see in Namapoikia. Now, one issue with all of this is that the scale of F. cooperi is much larger than Namapoikia. So if we have an issue with Namapoikia being too big to be a sponge, it's actually too small to be F. cooperi. So there are other potential options for Namapoikia. One idea is that we might be looking at something like Kinea, which is this wave-like, ripple-like fabric that you see in the Ediacaran. And people have done reconstructions where essentially if you have some sort of sticky substance, you can start to produce constructions that look at least superficially similar to Namapoikia. So one other question is, are we just seeing the effects of some sort of weird hydrodynamic regime built up over time? And this is something we can test by actually going to look at what reconstructions in Kinea look like in 3 dimensions. And I want to make the case that in general, microbial media constructions are functions of environment and biology. So this is an example from Slave Lake by Paul Hoffman, where you can see just the morphology of these stromatolites change depending on where you are in this regional setting. And so then it's not surprising that you might find thrombolites and stromatolites and something like Namapoikia being a putative microbial construction in the same reef system in Namibia. Okay, great. I have just about 2 minutes, and what I want to finish with is this point that new discoveries will continue to rewrite the record of life on our planet, and I'm looking forward to those discoveries. And then I'm just going to tell you where I'm going next. So I'm going to return to the archaeocyathids, and I told you at the middle part of this talk that Ryan Manzik has been working on archaeocyathids and trying to understand what their sort of 3-dimensional reconstructions look like and what they tell us about how they grew. And what Ryan has done in this paper is essentially produce 3D reconstructions of Cambrian archaeocyathids using Geary, and then compare the morphology of those archaeocyathids to modern corals reconstructed with X-ray CT. And Ryan has demonstrated that those branching angles that you see in the archaeocyathids are indicative of changes in, or indicative of the fact that these archaeocyathids can respond to changing paleo-environmental conditions. They have branching that is very similar to the sort of branching that we might see in modern framework reef builders. And so that really these are true framework reef builders, more so than the Cloudina. Once again, you're going to have to ask Ryan to really give a talk about these in detail. And finally, I just want to say that while I love the Ediacaran and I'm interested in continuing to work on them, so if anyone's interested in collaborating, I'm always available. I've actually turned my attention to microbial constructions much deeper in time. So this, for example, is a stromatolite from Great Slave Lake from the 1.9 billion year old Pathai group up in the Northwest Territories of Canada. And we've gone out there and done a bunch of field work, and I'm really interested in understanding if we can apply the same methods that I've been showing you to the reconstruction of these stromatolites to understand what the morphology is telling us about paleoenvironmental and paleobiological conditions, and whether there is some sort of diagnostic tool, although I know this is a long shot, for telling whether these laminated constructions are biotic or not. And so this is ongoing work, and in a few years, I hope to be telling you about that. And with that, I will open myself up for questions, and thank you for your time.
All right, thanks, Akshay. That was really cool. Can you hear me just fine? My internet's being kind of weird.
Oh yeah, no worries.
Okay, so awesome. We're gonna switch to chat discussion questions. You can raise your hand first. Find that raise hand button at the bottom of your screen. You click reaction. It's that first button. And if you'd rather type it, I can ask it for you. First one comes from Greg Vitalik. Go ahead, please.
Oh, that was wonderful. Can you hear me?
Yes, I can, Greg.
I think my video's on, but I can't see it myself. I agree with you that Namapoikia is something really odd, but I'm wondering there's another fossil that's been identified as Namapoikia in the red beds of Namibia. And I'm wondering which one is the type. The one in the red beds is a kind of a sheet-like thing that has holes in it. So I'm wondering, I'm not familiar with the taxonomy. Which is the real Namapoikia? These ones in the gray or the one in the one in the red?
Right, that's a good question. So a lot of the work that's been done is the Namapoikia that comes from Drudornvlakta farm. So it is the carbonate ones. Those are the ones that are in a lot of the descriptions. So in the original science paper on it and whatnot.
Oh, okay, good.
But that doesn't mean that there can't be other Namapoikia. Actually, one of the challenges here is that, I keep saying that it's only found in one reef setting, but that's just perhaps because we haven't been necessarily looking for it, right? So I would be interested in looking at these red beds, and there might be something very indicative about morphogenesis there or affinity that really can be proven by going outside of these carbonate reefs themselves.
Yeah, no, Great job. Thank you.
Thank you.
All right. So I guess I can ask, and maybe this is sort of an easy one. Sort of, I was going to ask for a little bit more. Maybe you can discuss some of the differences, like what you can get mineralogically versus morphologically, differences between this physical destructive method versus CT versus other non-destructive methods, because I'm very interested in some of this stuff. And I'm curious what the best approach is. I have some ideas. If there's different mineralogies that won't work and other things, I'm just curious to hear you talk about it a little bit.
Yeah, totally. The thing is, the first thing I would do with any sample is go do it in X-ray CT, because if you can reconstruct it in X-ray CT, it's relatively inexpensive and relatively quick to generate a really rough model to say, well, we do get responses or we don't. In those cases, when you do get a response, that's your reconstruction method of choice. People have been working on methods like ultrasound to do 3D reconstructions, and I think that works on much larger scales. So, if you're trying to look for crack structures through concrete, you're probably going to go in the ultrasound regime rather than the X-ray CT, rather than sort of grinding it away. That said, there are probably some really easy, basic rules to go by. If the density of everything in that sample, the density of all the features are similar. So for example, in the case of granites, you're probably going to struggle to do that 3D reconstruction with X-ray CT unless you're using these really interesting phase shift methods and what else. If, for example, a lot of the fossils I look at are carbonate and carbonate, so it's just everything's been replaced. You look at a thin section, it's all calcite that's been replaced. It's probably going to be really hard to get a three-dimensional form from X-ray CT. Not impossible, but it's probably a good indicator that you might end up going this destructive method. Everything that we do typically, I mean, even in the case of, I showed you that reconstruction of that calcareous sponge, we actually first imaged it in 3D in X-ray CT to demonstrate that it was worthwhile destroying it. So, many times, especially when these are one-offs, you want to be very careful. If you're going to go reconstruct ooids, there are enough ooids out there that you can do both methods and be pretty happy with them, right? Same with granites, same with actually some of these Ediacaran. Honestly, they're chock full and not many people are working on them necessarily. So.
Got it. Cool. Thank you.
Yeah.
So we don't have any hands up.
Maybe I can ask. I have a couple questions, maybe three. So first, you contrasted sort of animal build rift versus microbial build, and it's interesting contrast. But in stromatolite reefs, often you get very complex sort of distribution, and like you can have tidal channels. Just if you look on stromatolites, some of them kind of were eroded, so should be plenty of space for refuge and stuff like it. So maybe you can comment how it's important.
Yeah, so two things that jump off right off the bat. One is microbes play a major role in even modern reefs, right? So that's something I neglected to say, but microbes never truly go away. The question is a topographic relief from metazoans distinctly different? I think you've hit upon a really important point. So typically when people talk about reefs, they talk about the shift in morphology as you go from microbially dominant reefs to modern framework reefs. One thing I'm really interested in is whether that gross morphology actually does change. And I think we have to go look at things like surface roughness, right? So is the surface roughness of a modern reef, the structural complexity of it, truly distinct from a microbial reef? That's actually work that we're doing now by looking at these microbial reefs to really understand exactly what you're saying, which is when we see things like erosion, when we see towers built by microbes, are they on the millimeter to centimeter scale, even sub-meter scale, actually topographically distinct from framework reef systems? And I think my gut would be that they are, just because that branching tends to produce a completely different kind of roughness, but it's actually something that we need to test. There aren't many quantitative examples of that, right? So a dream scenario would be to say, yes, when we look at the surface roughness and the topographic relief of microbial reefs, they are, say, an order of magnitude lower than the ones that we see in metazoan reefs. Now, if it turns out that there is no difference, then we have to say, well, maybe they aren't really, that's not really the indicator of, that maybe reefs are always a cradle of evolution, and we don't need to wait for these frameworks to emerge. But I actually think that's work that needs to be done. A lot of it's been qualitatively described.
And in our question, you show that, well, John Gratzinger showed that it's restricted to Neptunian dikes, but I guess Neptunian dikes related to development of fallen basin in this area. But do you expect any hot fluids to go through them? The reason I ask, like, sort of, it comes in mind like this in early Archean, you get this chode dikes, so dikes with hydrothermal circulation, and you can have microbial mats living along the walls of this sort of voids, and so could it be like a microbial mats that form the structures?
That's a great question. I've never really thought about that. So I've always sort of hand waved the paleohydraulic and paleo environmental difference in those Neptunian dikes, but something I'd have to look into. But that is an interesting question. I mean, we know these Neptunian dikes are, they're occurring at the same time as the reefs growing. So Neptunian dikes are common in reef systems, right? As you get these breakoffs. Now, the question is there something else that's moving through them? That I don't have a good answer for, but it is an interesting idea. So it's something that we need to explore a bit more.
And the last question, just simple. So you showed different estimate for when sponges appeared, and maybe this estimate before Alice Turner paper was published. So I didn't see it included there. But my question, what is your favorite estimate for time when sponges appeared based on your work?
Yeah, so the molecular clock data tells us that they have to appear pretty early. So I do believe that. I think we are eventually going to find examples that are definitively sponge fabrics, and Turner's fabrics may actually be those, right? So I'm not saying that when I say they're disputed, I don't mean that I'm disputing them personally. I mean that these are disputed in general by the community. There are quite a few in there that might be sponges or might be something totally different. I think a lot of what Rob Riding's shown and others have shown in these consortia of stromatolites and sponges are really interesting. And so that's actually probably what we should be looking for, is whether those consortia, how far back in time those actually go. And one easy thing to do is compare the stuff that Liz has with the stuff that Rob has from the Cambrian, and to say, how similar are these actually in 3D, right? So are these the exact same fabric? In which case, then you might be able to really push that line to 890. If it turns out that those fabrics aren't similar, then I can tell you definitively by the Cambrian the sponges are around. Now, whether there's anything earlier, that's an open question. There are also spicule examples too. So, there are many examples of sponges. The big challenge here is, like, why aren't there more sponge fossils from the Precambrian, considering that they should have evolved early, and considering that, there are these really tantalizing signs of them. So my answer to that question is, I don't know. I can tell you that I know they're definitively there in the Cambrian, and that they're very, very likely there in the Neoproterozoic and the earlier end of the Neoproterozoic. So maybe those fossils that Turner talks about.
Thank you. I think there is one question in chat.
Yeah. Cool. Spencer Anderton asks, and Spencer, there's no dumb questions, but he asks, has Cloudina been found in situ, and is there any consensus on how it's attached to the substrate?
Right. So we often talk about Cloudina as having an apical end. There are many interesting papers that talk about how Cloudina was growing on a substrate, that you can see them actually sitting there recumbent on those substrates. My personal view is that, yeah, they're probably attached to something. We know we can see those apical ends. What that substrate is, it could be completely different. a lot of times when we see them in aggregate versus when we see them maybe say individually growing on a substrate, I think those aggregates might be more likely than not detrital. Personally, just from what I've seen and from what we've reconstructed. That doesn't mean that there aren't aggregates where they are in situ. And I know people have shown me different examples where they'd argue that they're in situ. And I come back to, okay, what we really need to see there, if we argue that they're in situ, are just signs that they're actually growing there, so that we see the apical end attachment, we see cementation that isn't just a single 2D cross-section. We also see that they have circular cross-section. So those are sort of in my mind, the diagnostics that we look for.
Okay.
Awesome.
Oh, it looks like we have a hand. Is that? Yeah, Paul, go ahead.
I'd like to come back to Darwin.
Yeah.
Darwin's expectation was that evolution was gradual, but the fossil record suggested it was stepwise. Where
Right.
are we now?
Yeah, it's a good question. I mean, you know, what's interesting is when we go to Ediacaran Cambrian, we're starting to talk more about a gradation, right? So we start to talk about these forms, these organisms making it through that boundary condition. You know, I still go towards this gradual form, and I come back to this question about what's happening with reef systems, what's happening to the morphology of reef systems. I expect that once we plot out the surface roughness of reefs, we're going to see this gradual transition as more and more metazoans start to occupy these reef systems and start to slightly change them. And so that rather than this being a step-like form, we're actually going to see some sort of gradual transition. That's where I am. I don't know if other people would argue strongly against me, but I would argue that one thing that we see often is the transitional nature of these forms, so that they do make it through what would traditionally be the Ediacaran boundary. A lot of times people talked about Cloudina going extinct at that boundary, and I think that's been challenged a bit as well.
Is that an expectation or an observation?
It's an expectation and an observation. On the transitional forms, that's an observation. On the stuff about reefs, that's my expectation. And so that's something we're testing.
All right. We've got a couple of questions from Kay Sender in the chat. He says, would 3D non-destructive imaging techniques aid a lot in CFD? And how would micro-CT and other non-destructive imaging techniques accommodate for diagenesis?
Yeah, those are both great questions. I know there's really interesting work coming out of Simon DeRook's lab, out of Jim Schiffbauer's lab, where they are doing 3D reconstructions, X-ray CT-based reconstructions, and then building models of aggregates, and then looking at computational fluid dynamics. So this is something that people are doing. They're doing to a lot of success. It helps us understand how these organisms may have fed, how their orientation may have been impacted by currents and vice versa. With respect to how micro-CT and non-destructive image techniques accommodate for diagenesis, there is a huge challenge when we think about diagenesis as being a set of, say, at least in carbonate systems, we think about maybe you have your initial porosity, it gets filled in with the secondary cement. That secondary cement tends to have material properties that are very similar to the initial rocks themselves. And so then we start to ask, well, can we actually make out various generations of cement from X-ray CT, from these non-destructive methods? I argue maybe in certain cases you can, if they are mineralogically distinct enough, or they are somehow materially distinct enough. But in other cases, when, for example, we're looking at modern ooids and we're trying to understand how multiple generations of cement may work on those ooids, you actually do need something that's optical or multispectral or even geochemical to really tell the difference between those. And so those are places where the ooid itself and the secondary cements might actually look exactly the same in CT.
Okay, we don't have any hands up right now.
Or maybe just a quick question. So you mentioned why we don't have more of an evidence for sponges in Neoproterozoic, and I always wondered, could it be that sponges evolved, but where morphological sort of expression evolved later? And related to it, did you try to do any work on spicules? Because it seems like with your method, it can add something in terms of three-dimensional and internal structure.
Yeah, so I've not worked on spicules, but that is an interesting area of study. Shuai Zhao's lab is doing a lot of this work. It is an interesting question about the gross morphology of sponges. We know that there are certain traits that sponges have to hold, right? The canal systems, you can't get rid of the canal system and still call it a sponge, but whether you can expect to see calcareous sponges that early on, whether the spicules should have similar morphologies to the spicules you see later on, or whether they start with some weird precursor and they're maybe not as developed. Those are all open questions, I think. That would explain why we might not see evidence for those. There are other geochemical reasons why those spicules made out of silica might just dissolve away, and so they're rarely preserved. I think what would be interesting is if those keratose fabrics that you see are similar to the ones that you see in the Cambrian, at least then you're starting to say, okay, those fabrics seem to exist. So there is some continuity there, which would then still raise the question, if there's continuity across, 200 million years or 100 million years, why aren't there more other putative sponges in that record, right? So why do we have a dearth of those sponges? Calcareous sponges probably evolved later than silicious ones. And so that might be okay that the only time we see calcareous sponges is right in the Precambrian, right? Right running up right into, but silicious ones might or should appear earlier. So yeah, those are great questions. I don't have good answers for them.
Thank you.
Okay, I'll give
Awesome.
a couple seconds in case someone wants to speak up.
How do I do that?
Oh, is Daryl? Are you want?
Yeah, I
Go
just
ahead.
want to make a comment about the lack of sponges. I think a lot of it is diagenesis. Liz's stuff, she found it in amongst about 300 or so thin sections. So, and there's only one occurrence. And it was really difficult to see it even in that. And, it would be lovely to do CT on it, but there's none of the sample. Like when you make a thin section, you've taken the whole damn thing. So I think it's probably because most Precambrian geologists do not do enough petrography in the first place to look for them. They're looking for form fossils and form features, and they're not being totally myopic, which they should be.
I absolutely agree. And, I think so I totally agree. I think a lot of times we just have to look at the rocks and realize that they've gone through a whole bunch, right? So as soon as you look at that thin section, I mean, like I was saying, a lot of these when we get to the Precambrian, we look at the Cloudina, everything's replaced in that, right? So you can look at that thin section, you can see that those are not original calcite or aragonite or anything in there, right? And so then it becomes a real challenge when we go further back in time, like you're saying, diagenesis is going to erase a lot of those signals. And I 100% agree that if you're finding this in a single thin section, it becomes really hard to evaluate whether or not that thing is actually what we're saying it is. So that's work that needs to be done.
All right. Let's see. It looks like everybody's pretty satisfied. So that was great. I really enjoyed that and good discussion. So thanks a bunch, Akshay.
Thank you all.
Best of luck continuing this type of work.
Awesome.
And everybody take care. We'll see you again next week.